Antibodies are little proteins that are found in the plasma part of whole blood. The official word for antibodies is immunoglobulins, and this term reflects the fact that these proteins are measured as part of the gamma globulin proteins that can be measured in plasma. The heavy chain is a sequence of about 400 amino acids. Heavy chains are connected together by disulfide bonds, which are similar to the bonds that connect nucleotides in DNA. The heavy chains form the core of the Y-shaped immunoglobulin. There are binding sites for complement proteins on the trunk or the heavy chain of Y-shaped proteins. And the heavy chains come in 5 flavors: M, A, D, G, and E. The constant region of heavy chains does not change among immunoglobulins of the same class. So all M immunoglobulins have the same heavy chain constant region. The variable region of the heavy chain binds to specific antigens and has an antigen binding site at the very tip of it. This allows an immunoglobulin to bind to antigens inserted in the plasma membranes of pathogens. Light chains are the smaller chains, about 200 amino acids long, and on the outside of the heavy chains. There are two different light chains, kappa and lambda, and just as with the heavy chain, the constant region is conserved within a class of immunoglobulins, and the variable region is capable of binding with a specific antigen. A immunoglobulins are common in breast milk and mucus. Conditions such as the flu and COVID would increase A immunoglobulins in your respiratory mucous membrane. All 4 mucous membranes of the body, reproductive, urinary, digestive, and respiratory, have a coating of mucus between the cells and the lumen. That coating of mucus is rich in A immunoglobulins. G immunoglobulins are significant in that they cross the placenta and they confer passive natural immunity to baby from mom. If you are of a negative blood type, you make G immunoglobulins that like to attach to the Rh antigens that are embedded in the cells of somebody with a positive blood type. RhoGAM is a shot given to blood type negative women, usually upon their second child. What happens is that during the first birth, mom can get exposed to baby's blood, and then mom starts to make the G immunoglobulins that attach the Rh protein in blood type positive cells. So therefore, during the second pregnancy, if blood type negative mom is pregnant with a blood type positive baby, mom will have immunoglobulins capable of attacking the cells of the Rh positive baby. RhoGAM prevents that attack. E immunoglobulins are common in basophils and mast cells. So allergies. Basophils circulate through your blood vessels while mast cells take up residence in tissues. If you encounter an allergen to which you are allergic, for example, I am allergic to the penicillium fungus that makes blue cheese. If you're exposed to it, you will begin to make E immunoglobulins. That's not terrible. But these immunoglobulins, they'll get inserted into cell membranes of basophils floating in your blood and mast cells residing in your tissues. Then, upon second contact with the allergen, so the second time I ate blue cheese, the blue cheese antigens connected with the E immunoglobulins inserted into the basophils and mast cells. And when I say they connected to the E immunoglobulins, I mean they connected to all of them. This massive connection of antigen and antibody triggers a systemic release of histamine, producing anaphylaxis. The last two immunoglobulins are commonly inserted into B cells, which are the cells responsible for initiating the process of making immunoglobulins. So, which event happened first? I don't know. But B cells that experience insertion into with B cells that experience the insertion of immunoglobulins are then ready or primed to act quickly if the antigen is detected a second time. For example, many vaccinations expose your body to attenuated viruses or viruses that have been chopped up into tiny little pieces and therefore cannot really make you sick. But these tiny little pieces of the virus are capable of provoking the production of M and D immunoglobulins. Therefore, the second time you encounter whatever it was you were vaccinated against, those M and D immunoglobulins will connect with the antigen and they'll speed up your immunological response, amplifying it compared to the first response. This picture here, it doesn't do the M immunoglobulin justice. It usually is in a pentagon shape or a five-pointed star shape. It's a very, very large immunoglobulin. Immunoglobulin production begins with stem cells in the bone marrow, and we came to find out that there are also stem cells that produce immunoglobulins in the liver. Hematopoietic stem cells differentiate into myeloid and lymphoid stem cells. And then those lymphoid stem cells can become various types of lymphocytes, B cells, T cells, or natural killer cells. Once a lymphoid stem cell becomes a B cell, it's like preloaded with immunoglobulins determined by genetics. You have a predetermined set of genes that make immunoglobulins. So you have these B cells in your lymph nodes that just kind of hang out and wait for their antigen to show up. And to be honest, they're not primed for one antigen, they're primed for many. So over time, if the body encounters the antigen, it will bind to the immunoglobulins on the B cells, and then the B cell will display it to T helper cells. When this happens, the B cell signals the T cells for help and it starts cloning itself. These clones of the B cells, the right type of B cell to make the right type of immunoglobulin, these clones create plasma cells. It's plasma cells that produce immunoglobulins. Not B cells directly. B cells do mitosis to become a plasma cell, and then the plasma cell starts pumping out immunoglobulins. Additionally, the B cell will differentiate into memory cells. And those memory cells will store the information about the antibody for quicker responses in future encounters with the same antigen. That is immunity, having memory. So while the initial response to an antigen involves all of these steps, memory cells expedite the process and they allow for a much faster and possibly much more effective immune reaction upon second exposure. The antibody production process is relevant to concepts like the RhoGAM shot. Like, for instance, if a person with O negative blood carries an Rh positive baby, their body may not initially produce the immunoglobulins against the Rh antigens. So, this is why no RhoGAM is needed for the first baby. But, as I said previously, during delivery, mom's blood can be exposed to baby's blood. And now mom starts to make those Rh immunoglobulins. So the second blood type positive baby that mom carries could be attacked by those immunoglobulins. Immunoglobulins play several roles in pathogen elimination. They don't eliminate a pathogen directly, but they help in its elimination. As we saw back in blood typing, immunoglobulins and antigens can link up and cause agglutination or clotting. Then the clumps are phagocytosed. Immunoglobulins can precipitate antigens from solution, kind of like undissolving them. The linkage of antibody and antigen forms this big like lattice or network, and that basically becomes undissolved. And just as in agglutination, the clumps are phagocytosed. Immunoglobulins can also assist in cell lysis. Antibodies smothering a pathogen can provide a place where complement proteins can fix themselves and do one of those MAC attacks. This was where the complement proteins assembled to make a hole in the pathogen cell membrane, and then it caused the pathogen to lyse or split. Immunoglobulins can smother pathogens by binding to their surface, and this is what is called opsonization. This term refers to antibodies or complement proteins covering an invader. For instance, a pathogen covered in antibodies can lead to agglutination or precipitation. And essentially, opsonization makes the pathogen more attractive to macrophages, which, I mean, they see it as like tasty. The root word for opsonization means condiments, like ketchup. And just as ketchup makes fries much more appealing, antibodies covering a pathogen make it more attractive to macrophages. Okay, so now all of these types of marking antibodies and marking antigens can lead to the destruction of the membrane-bound antigen. We can attract phagocytes and then they can engulf the antigen. We can use cytokines to get macrophages and basophils to come to the area, neutrophils as well, promoting them to release histamines and trigger an inflammatory response to the antigen.